Thank you. So thanks for inviting us to speak today about CANCOV. So Margaret, Herridge, and I will actually do a tag team approach to this presentation. So as I was saying, that Margaret and I will do a tag team on this presentation. So CANCOV is a Canadian multi-center COVID-19 prospective cohort study with very detailed longitudinal one-year follow-up of patients in BC, Alberta, Ontario, and Quebec. And we are proposing to recruit 1,000 hospitalized patients and 1,000 non-hospitalized patients, and also 500 caregivers. There are more than 100 investigators across Canada involved in this study. So the purpose of it is really trying to establish a patient-centered platform to collect clinical data, research data, and also biospecimen, really to support the interdisciplinary network of experienced investigators and junior investigators to answer urgent scientific questions related to COVID-19 infections. And these activities include host and viral genetic profiling, serological and immune testing, epigenomics and transcriptomics investigations, as well as collecting very detailed short and long-term outcomes and complications of these patients. So we're trying to recruit the patients at baseline when they have the infection or when they are hospitalized, and then sort of collect data at the short term within the hospital stay or within 7 days, and then 1 month, 3 months, 6 months, and 12 months. And we're collecting social demographic data and comorbid illness and medications and symptoms, quality of life, complications and function, and also healthcare utilization. And what we're doing is really trying to provide a mechanism to avoid the development of silos of small and large data sets, and trying to realize the full value of investments that have been made in COVID research in Canada. So our overall objective is really to determine short and longer term outcomes in patients and their caregivers, and also the clinical, social demographic, and genetic, and it's actually multi-omic predictors of these outcomes. So originally, when we applied to CIHR, we proposed 5 projects: genetic profiling, the multi-omic biomarkers, and also short and longer term outcomes, and using AI to really better understand the predictors of poor outcomes. And each of these are led by different people. But it actually quickly involved a number of other studies. So all the highlighted ones are supported studies by CIHR. So, you know, there's a Recover project that Margaret has been leading. And it's a transitioning care project in the ICU population. There's a PODS that Karen Okranik has CIHR funding that looking at transitions in care in general internal medicine ward patients. And also the Gemini Network, which is general internal medicine, the inpatient initiative. And this is the hospital analytics lab. But it quickly expanded to involving the COVID-free at home, which is a remote monitoring of early symptoms to see if we can make a difference in the outcomes, led by Robert Wu. And there's a piece that is on social and structural determinants of health, as well as looking at the illness experience of racialized patients, which is led by Shale Rawal. There have been a number of biomarkers kind of studies, including the cyclic study, which is led by SickKids PI. It's the cytokine, complement, and vascular endothelial injury study. And Beacons, which is actually led by PIs Paul Kim at McMaster University, which is on coagulation factors, and GenCov, which is led by PIs at Sinai, looking at viral and host sequencing and also antibody response. And there are many other studies. I won't have time to go into each one, but we are now sort of an umbrella for some of these other studies looking at muscle, looking at neurocognitive study, and linking also the contacts of these COVID positive patients to something that's led by Daryl Tan, COVID PrEP. There are quite a number of studies that are sort of under this umbrella. I want to just give you a little sense of where things are at. So at University Health Network, we started recruiting in August. And currently, up to today, we have 168 participants. And 77 are non-hospitalized cohort. In our non-hospitalized cohort, 18 of them are actually non-ICU, and then 26 of them are ICU in the ICU cohort. And you can see the corresponding number of caregivers as well. Now, this is actually a, people talk about adaptive trial. This is actually an adaptive cohort study. One thing we learned was that we have a whole bunch. When we first opened, we have emails from 400 people saying that they have long COVID, or actually they call themselves long haulers. And so we changed our protocol to include a group that is using antibody testing because they didn't test positive or they didn't get a test initially. And so that group, we have recruited about 37 of them right now. And so we are changing the protocol depending on sort of what we learned as well. So this slide is actually initially published earlier this year in Nature Medicine, looking at all the extrapulmonary effects of COVID-19. What we don't really know is how often these things occur. I can tell you that we have one patient with PE, we have one patient, so I think it's still early to actually get a sense of these symptoms. One thing that is common is that most of the patients that we've seen, it may be because of how we test patients as well, using nasal swab test, most of them. They present with respiratory symptoms. And at the 6-month point, I can tell you that a lot of them still have some post-viral cough, and some, there's a large number of patients with fatigue, even in the non-hospitalized patients, and also muscle weakness and brain fog. And one interesting thing is, even for the loss of taste and smell, as people are regaining some of those, we hear very interesting stories about patients just, smelling chicken noodle soup everywhere. So I have one patient coming in saying that to me and saying, I smell chicken noodle soup everywhere. And some are saying, their sense of smell, yes, they're regaining it, but it is a mix of peanut butter and stale coffee, or it smells like smoke. So some of these stories are quite interesting. And now I'm going to hand it over to Margaret to talk about the ICU crowd.
Thanks, Angela. Angela's going to advance the slides for me. So I just wanted to show you a picture. Actually, this patient is in our other Recover study, but just to give you a sense of what almost the entire ICU here at the Toronto General looked like in the first wave, where our entire census was COVID patients, many of whom were on ECMO. You see the ECMO machine here off to the patient's right, on renal replacement therapy on a vent. I mean, this is about as sick as you can get. And our whole unit looked like this. And maybe next slide, please, Angela. Just to share a couple of things that Angela and I worked together on our 5-year ARDS follow-up study. And I will say that we use the basic platform of this cohort to inform, again, the ICU cohort of CANCOV. And already I would just say anecdotally that the post-critically ill COVID-19 patients are extraordinarily reminiscent of the post-ARDS patients from just general ARDS risk factors, in that they have remarkable muscle wasting and weakness. And you can see here on the top picture of someone we did a home visit on at 3 months, typically losing about 20% of their lean muscle mass. And here, an early muscle, some early muscle histology consistent with the myopathy. Here you can see angulated, sort of multinucleated muscle fibers, specifically a myosin depletion myopathy. So just the ARDS piece looks extremely reminiscent. Next slide. And I would also highlight that in our ARDS study from many years ago now, because of the functional disability, patients had a decrease in 6-minute walk distance. Here you see at 1 year, patients, and this was a young cohort of people in their early 40s with very severe ARDS. 40% of this cohort died. So a very high mortality, similar to COVID-19 when people became critically ill with ARDS. Able to walk only 66% of an age and sex-matched control population. And strikingly, having really quite preserved pulmonary function. So again, our early follow-up in the ICU cohort, which has actually mostly been in person. I personally have seen the majority of these patients to date with our team. A very similar reduction in functional outcome, muscle wasting, and remarkable preservation in the survivors of pulmonary function to date. Again, anecdotal, but those seems becoming more robust, but we'll need to look at the entire cohort, obviously, before we can make any conclusive statements. Next slide. So just to explain that it's very hard, to bring studies up de novo. The ICU arm of our CANCOV program is built on ICU follow-up program called the Recover program. And next slide, please, Angela. And this was an already CIHR funded study underway. First phase had been completed. That was a 10-year study across Canada of looking at outcomes in patients and caregivers after a week of mechanical ventilation, really designed to inform a continuum of care pathway for post-ICU patients and families. And our second phase, also CIHR funded by a Transitions in Care grant, was underway with 20 patient caregiver dyads to pilot this continuum of care pathway when COVID struck. So we halted that study and we really built the ICU arm of CANCOV on the Recover program platform. And I think many other groups did that as well to try to speed up the process, just to highlight that we did that here. Some clinic photographs already you might find of interest. Obviously, cannulation on ECLS unfortunately leaves patients with quite significant scarring. Here you see the typical configuration would be an IJ, right femoral cannulation, and here you see the resolving scarring of the right IJ cannulation in the trach. And a patient who was really wanted to share a lot of photos with us, and you can see here the vasculitic rash of COVID. This is a very young patient who actually was never intubated on high flow oxygen in the unit because she was just on the cusp of needing an airway, but unfortunately had likely a CNS vasculitic manifestation and did have a stroke and still had mild left-sided hemi-weakness when I saw her in clinic. Next slide. So a lot of the physical morbidities we see in the post-ICU patients, obviously we see here in the post-COVID-19 patients, and I list them all here. I might indicate that just being a very severe critically ill patient does cause changes with taste in particular, perhaps not so much with smell, but people have multi-system challenges, and this will be one of the challenges to tease out what's really related to ARDS, what's related to critical illness, what's related to being in the ICU for a very long period of time, and what's specific to COVID-19. And these will be some of the challenges that we will, interrogate our cohort data set for. Next slide, please. One thing that we've really emphasized in all of our post-ICU studies is mental health. Post-ICU patients and family caregivers have very important, often severe mood disorders or exacerbation of pre-existing mood disorders. This is increasingly thought to be related to injury, a structural injury to the amygdala. So there's a structural correlate for the very severe mood disorders we see, exacerbated by a very, very heavy drug exposure in the ICU with sedation, narcotics, a complete derangement of the circadian rhythm. These patients have profound circadian dysrhythmia and are profoundly sleep deprived. The average ICU patient only sleeps for a few hours every night, and there's a complete derangement of normal sleep architecture. So it's not surprising that people have a lot of mental health issues, but both patients and caregivers felt to be on the basis of an unrelenting hypercortisolemia and a similar damage to the amygdala. In clinic, we've had a lot of mental health resource and interrogation, and a lot of I've listed them here, but in the interests of time, challenges with anxiety, with depressive symptoms, with PTSD. It's very tricky for patients because COVID is in the news cycle 24/7. The physical injury obviously can trigger anxiety. Delirium is a huge issue with the COVID-19 patients, and often people lay down traumatic memories from delusions from ICU delirium. Often these are persecutory delusions relating to PTSD. You see a slice of reality here completely distorted. Aliens in yellow would be all of us in these yellow isolation gowns. Often violent images, being on a boat because you're on an airbed, these sorts of misperceptions. A lot of people very disturbed by global cognitive dysfunction, the so-called brain fog, but also having specific issues like anomia, agoraphobia, feeling completely transformed by this experience. But also some people articulating an enormous sense of gratitude and also a willingness to be in the study because they want to participate, educate. It's a way of giving back to groups who they feel grateful to. Next slide. And just to highlight caregiver issues, this is some of the data from our Recover program and that I led with my co-PI, Jill Cameron. Just trying to illustrate here that caregiver mental health issues should not be minimized at all. And they're usually new, but again, they can be an exacerbation of pre-existing mood disorder issues. I'll show you here at 7 days, 53% of caregivers have moderate to severe depressive symptoms. And this persists to 1 year. 27% of the caregivers in this mechanical ventilation cohort that Jill and I led had moderate to severe depressive symptoms. And some caregivers in this cohort reported suicidal ideation, and there was one suicide attempt among the caregivers in this cohort. So not to be minimized, the impact on family, not just the patient, and that's why we've tried to bring a family-centered approach to CANCOV. Next slide, please. So just to highlight again, some of our co-PIs, and we had co-PIs across different aspects of the study, across different provinces, all listed here. And this was the way Angela and I organized the study because we have an enormous number of collaborators now under this CANCOV consortium umbrella. and in collaboration with pre-existing research groups. Our own Recover group, we're indebted to the Gemini group, and we collaborate with the Canadian Critical Care Trials group and the CCIR-NET investigators, which is a community critical care Canadian research group. Next slide, please. So a lot of people, as Angela said, we have over 100 investigators, and I will say it's ever growing. So many GIM investigators, critical care investigators, and across very varied disciplines, psychiatry, transplant, neurology, and in rehab, we have a lot of support from our biostatistics, from the BRU here, our health economics and technology folks. Next slide. Our rehab collaborators at Toronto Grace, collaborators at Sinai, St. Mike's, Sunnybrook, the Gemini Network. We're grateful to Fahad Razak and Amal Verma for their kind collaboration here. Jane Batt, Claudia DeSantos, also co-PIs on the basic science. Lisa Strug and Ray Young, co-PIs on the genetics piece, and many Sick Kids colleagues. Next slide, please. And community partners. This is really also, Angela and I've really tried to emphasize the importance of working closely with all of our community partners, and so many community partners saw so many COVID-19 patients. So community partners at Trillium, Markham Stovall, Ali Binney, CCIR-NET co-PI. She with Jen Sang are leading the Canadian Critical Care Research Network for community intensivists. Michael Guerin and Wes Park, our colleagues there. Computer Science and Medicine Vector Institute. Marzier Gassemi, the PI there. Patient and Family Engagement Representative Cheryl Misak, PTOT, Biomedical Engineering. Next slide, please. And other Canadian sites, London at Western, Kingston at Queen's, St. Joe's in Hamilton, Ottawa Hospital, University of Waterloo. Next slide, please. And our Quebec collaborators in Montreal and in Sherbrooke, Suzanne Morin and François Lamontagne are the Quebec PIs. Our Alberta collaborators, the Alberta PIs, Sophia Ahmed and Tom Stelfox, collaborators in Winnipeg, Saskatoon, and British Columbia, our BC site PI, Anita Pelapoo. Next slide, please. And then many folks across the Canadian Critical Care Trials Group, the Canadian Critical Care Society, a lot of different critical care partnerships that we're very grateful to. Next slide, please. We want to say thank you to all of the groups that have funded us. Like many groups, some of us are using our current CIHR funds to help support and cobble together ongoing funding. Angela is using some of her funds. Our group is using some of our Recover funds. We're very grateful to the U of T for their generous donation to this, generous support to the study, to CIHR, to the Canadian Frailty Network, and to the COVID-19 Immunity Task Force. Like many groups cobbling together many funding sources to bring a larger enterprise together. Next slide, please. And just to end here, we're trying to work more and more on our website, allowing patients and families to reach out to us to promote the cohort across Canada, and hope to get as many folks engaged as possible. So we really have a wonderfully generalizable sample of patients and caregivers across a spectrum of illness for COVID-19. And I think that's the last slide, Angela, is it?
Yeah. Yes.
Yeah, and I'll just finish by saying, you know, it's a pleasure to work with Angela. She's really the mastermind of all of this networking to create this massive CANCOV consortium. And I just want to acknowledge her important contribution in that way. Thank you very much.
All right. Thank you very much for that presentation. It's really an incredible effort to take this holistic approach to really tracking the natural history and outcomes of COVID in the Canadian population. I mean, we're a bit over time with the talk, but I do want to just ask one question because it is a monumental amount of data that you have coming in. Is this going to be centralized at UHN, this sort of data repository? Are there going to be access for researchers to some of this?
Yeah, I mean, we can both speak to that. I think that our mission here, at least from the clinical side, is to create really the definitive, extremely granular, multi-dimensional characterization of the patient phenotype across the different cohorts representing different spectra of illness. That resides here, that's right. And I don't know if Angela may want to speak to some of the biological collaborations, but we're really trying to put a lot of these pieces together that Angela began the discussion with. Angela, did you want to comment on that as well?
For sure. We are taking a bit of an open science approach to this by asking people who are actually have grants to support some of these analyses to contribute back the data so that other people can ask other questions. We do hope that, other people will want to ask questions as well.